Juvenile white sharks form aggregations in Southern California. These are large groups of up to 40 sharks that stay at a relatively small area for months on end. Because there can be so many sharks in a small area for a really long time, we expect that these aggregations are having some sort of effect on the broader prey community. We have a pretty good sense of juvenile white shark diet from prior research. For example, we know that benthic elasmobranchs, which includes things like stingrays, smaller sharks, and guitarfish, make up about a quarter of the white shark diet. What we don't know is the type of ecological effect these aggregations are having on the prey community. To answer some of these questions, we turn to a technology called eDNA metabarcoding. eDNA is short for environmental DNA, and it just refers to any loose genomic material in the environment. Metabarcoding refers to the ability to take all the sequences in a sample and match them to the species that they came from. What we did was take surface seawater samples from 3 juvenile white shark aggregation sites and 2 non-aggregation sites as controls, and extracted all the DNA out of those water samples. We then sequenced the DNA and matched each sequence to the species that it came from. By doing so, we get a sense of what species are present at a site, as well as a rough abundance estimate of those species. So we get a good sense of what the community structure looks like at all of these sites. What we found was that the more a prey group contributed to the juvenile white shark diet, the greater the decrease in that group from a non-aggregation site to an aggregation site. For example, benthic elasmobranchs make up about a quarter of the juvenile white shark diet. That is the most targeted prey group, and it decreased by 75% at aggregation sites compared to non-aggregation sites. This relationship between contribution to diet and change in abundance between controls and aggregations was pretty strong. The proportion of diet each prey group contributed explained about 60% of the variation in that change between control sites and our aggregation sites. On the other hand, prey groups that were not targeted by white sharks, for example, forage fish, large and medium pelagic fish, which each made up less than 3% of the diet, actually increased at the aggregation sites. These species may benefit from the suppression of mid-level predators. Our results demonstrate that it really is dangerous being small and flat like a pancake. Our results will also help lifeguards understand the ecology of the juvenile white shark aggregations at their beaches and help us coexist with these incredible